I'm really happy to be with you tonight. Hello, good ladies and gentlemen. So we will speak about taste, and more particularly about taste of wine. Indeed, taste is a very common word used in our daily life, and it has various definitions. Taste can be, according to Cambridge Dictionary, taste can be the flavor of something as in your mouth, the ability to sense different flavors in your mouth, but also the act of trying something to find out what flavor it has, and the ability to judge whether a piece of music, art, fashion is good or bad. Indeed, when you have something in your mouth, you have taste. We will see why. But you have not only taste, you have also touch by contact with your mouth, by astringency, for instance, precipitation of protein due to tannins, for example. And you have another stimulation. You have, due to retroolfaction, you have also a smell stimulation. And so there is a kind of confusion between aroma and taste. When we speak about cork taint in French, we say good bouchon, that is a cork taste, but it is not a taste, it is a smell. But due to the location of the taste and smell perceptions, we can have a confusion. In this talk, I will speak about the taste in the strict sense, and we will focus on the taste perception. How can we perceive taste? We can perceive taste due to receptors. These receptors are located on some structure in cells that form some bud taste. And this bud taste, you can see the bud taste on the right part. And on the left part, you can see that the bud taste are inserted in papillae. And three kinds of papillae, circumvallate, fungiform, and foliate papillae, carry this kind of receptors. And you have interaction like a kind of key and lock, and you have some stimulation of the receptors, and then a nervous signal sent to the brain. This kind of bud taste will carry various receptors that help us to perceive the various tastes. We have commonly, we can distinguish 5 basic tastes that are sweetness, sourness, bitterness, saltiness, and also umami taste. That is less common maybe in European feeding, but that is very important for Asiatic people. But also, if we look at this particular taste, it is also very present in our feeding. For instance, in meat, in the soup, you can have a lot of umami taste. But this distinction between the 5 basic tastes is a bit extremist, because we have, like for the primary colors, we have a lot of shades. And for instance, some compounds like aspartame is mainly sweet, but it can also develop a bitter taste. And it's the same. We can see that malic acid doesn't develop exactly the same taste as lactic acid or tartaric acid, but they are described as sour, and there is a loss of vocabulary. For instance, Aristotle used more than 10 flavors. I think it was 14 flavors to describe the taste, and today we have 4 or 5 words, so it's a bit poor. And how can we perceive the various states? By receptors, and these receptors explain why we have a lot of various states according to the molecules. We have one receptor for sourness, one for saltiness, one for umami taste, one for sweetness, but with various mechanisms of interaction with various binding sites. And more particularly for bitterness, we have 25 receptors that is linked to our our diet because, for instance, some animals that eat only meat have less receptors to bitterness, and humans are a good equipment to perceive bitterness because they can eat some vegetables. Some of them develop toxic compounds with bitterness. So we have a huge equipment to perceive taste, and we will focus more about the taste of wine. Philippe Darrier and Alexandre Pons will show you how the aromatic typicality of wine is a key point for wine quality. But gustatory balance is also very important because this gustatory balance will rely on sourness and sweetness in white wines, but also on sweetness, sourness, and bitterness in red wines. That are the definitions of these balances by Émile Pénaud in Le Goût du Vin, another kind of taste. And if we look at this taste on a molecular point of view, we can see that the explanation can vary a bit according to the sourness, the accuracy of the explanation. We well understand the sourness because the organic acids present coming from grapes, for instance, like tartaric acid, malic acid, and citric acid, or coming from fermentations or on the microorganisms activity, like acetic acid, lactic acid, and succinic acid are well known. So we have a good understanding of the origin of sourness in wine. Concerning sweet taste in sweet wines with residual sugars is not difficult to understand. It is due to these residual sugars, that means fructose and glucose, that are still present after the end of the alcoholic fermentation because they were at high concentration in the grapes, for instance, or maybe because the fermentation has been stopped. But for dry wines, that means with residual sugars below their detection threshold, it's much more difficult to understand why they develop a kind of sweetness. And if you taste a solution, I think maybe Jane remembers that she tasted some solutions of ethanol with glycerol and with tartaric acidity, malic acid, and the perception of acidity, of sourness, and bitterness is very high in such solution. Why? Because it misses something that can balance the acidity. Some people can claim that ethanol and glycerol are responsible for the sweetness in dry wines. But in various experiments, this topic was discussed, and we demonstrated that the variations in ethanol and glycerol content in dry wines are not responsible for variations in sweet taste. So ethanol and glycerol are not cannot explain the sweetness in dry wines. They are something other, and I will try to give you some illustrations. Finally, concerning bitterness, we know a lot of compounds that are the tannins that can develop bitterness, but it's not so obvious because when we speak about tannins, we don't know exactly which compounds at which detection threshold are responsible for bitterness. So for this flavor also, there is a lot of work to do. Professor Denis Dubourdieu defines the winemaking as for him, winemaking consists in guiding the natural phenomenon of the transformation of grapes into wine, intervening as little as possible, but wisely. And I really like this definition of winemaking because it illustrates the importance of the elegancy of the conception of winemaking and the importance of understanding the mechanisms of this transformation to be able to act as less as possible but wisely. You know, when you have no knowledge, you have the choice to do nothing or too much. and the good knowledge can help you to have a wise winemaking process. So to study the origin of sweetness in rye wines, during the PhD of Blandine Crétin that has been defended in 2016, we made some experiments with alcoholic fermentation in various tanks from Merlot and Cabernet Sauvignon. And at the end of the alcoholic fermentations, 3 barrels were run off, and they underwent malolactic fermentation to give modality AF for alcoholic fermentation. And the tank was carried out the post-fermentation maceration, the traditional post-fermentation maceration that is used in Bordeaux, but also in Burgundy and in various vineyards during 10 days at 30 degrees. And after the end of this maceration, 3 barrels, same kind of barrels, were run off and underwent malolactic fermentation to give the modality PFM. So you have the wine with the modality PFM as it is, and the wine as it would have been if he hadn't undergone the PFM. So it was interesting to compare these 2 modalities. If we look at what occurs during post-fermentation macerations, we have 2 phenomena. The first one is yeast autolysis, because due to high temperature and the end of the alcoholic fermentations, yeast are autolyzed during this period. And we have also contact with solid parts of the grapes, with the skins, with the seeds, and sometimes for some kind of winemakings with the stems. And first, we compare the modality AF and PFM. And for modality EF, there was no autolysis, no contact with solid grapes, and modality PFM at both. And we can see that there is a significant difference of sweetness, with more sweetness in the modality PFM. That means that sweetness, we can demonstrate what winemakers currently observe, that is, sweetness increases during PFM of red wines. We tried to give an explanation of this phenomenon, and for that, we made a modality EF plus Y by adding some yeast in the modality EF. That was a dry wine, so without any sugar, so the yeast were autolyzed directly in the wine. It was all the more that it was kept at 30 degrees during 10 days. So we obtained a modality AF plus Y with yeast autolysis and without contact with solid parts of the grape. So we can observe what was the yeast effect, the autolysis effect. It was a collaboration with Virginie Moine from BioLaffort, and we observed significant differences of sweetness intensity between these 2 modalities. And we had more sweetness in the modality with yeast autolysis. We gave an explanation of this phenomenon, of this observation, and we demonstrated. I will not show you all the results because it would be too long, but we demonstrated that there was a key role of a protein, HSP12, that is a protein to respond to stress and to heat stress, and the release of some peptides coming from these proteins can increase the sweetness. During the PhD of Antoine Léger, he showed, under the supervision of my colleague Charlotte Cabane, he showed that there was variations of HSP12 content according to the strain. So, you know, the strain can influence the aroma of wine. But here we demonstrated that the strain can also have an influence of the taste, on the taste of wine. And some strain can give more sweetness to wines than others. It's important because we have to, enology, as Denis Dubourdieu said, is understand to act. And it's important to give an explanation of why some sweet wines develop some sweetness. That is autolysis of yeast proteins. but it's also important to have some applications. Then we compare the modality AF plus Y and the modality PFM. The difference was not autolysis because both underwent yeast autolysis. The difference was contact with solid grapes. And again, we observed significant differences. That means that the effect observed, the increase of sweetness observed during PFM is due on one part to yeast autolysis and on the other part to grape effect. And so that suggests the release of sweet compounds that are not sugars and that are present in the solid parts of the grape. A part of the PhD of Blandine was to investigate the nature of this compound. And she used a procedure, a purification protocol that was taste guided. We use various separation techniques, and after each step, the fractions obtained were tasted and submitted to sensory analysis to try to identify which one were the sweetest. And the sweetest were kept for the next step until we obtain a pure compounds. In this way, Blandine purified two compounds coming from grapes with a sweet taste. This one, EPDPAG, and this one, astilbene. Astilbene is a phenolic compound, but it has a sweet taste. Marie Lescanf will speak you more about astilbene a bit later. For applications, we had a comparison, an interesting comparison for some tanks of Merlot in Pauillac, and we compare low extraction and high extraction, that is by double the pumping over volumes. And we observe that paradoxically, the level of EPDPAG and astilbene, the sweet compounds, were lower when the extraction was higher. Maybe there is some degradations or maybe some bindings of these compounds on the solid part, rebinding of these compounds on the solid parts. We can have various explanations, but it's very interesting to see that when you extract less, you have more sweetness in the wine. Another example will be was an experiment we made some years ago with Pinot Noir in Burgundy, and we compare winemaking with destemmed grapes and in whole grapes, and we observed that there was no difference for EPDPAG, but for astilbene we had more astilbene in wine with whole grapes, and it was due to the high presence of this astilbene in the stems. So it's also a part of the explanation. Be careful, because I don't mean that you had to use stems everywhere to increase the sweetness. No, when the stems are ripe, and for some varieties, it can be interesting, and it can explain why this Pinot Noir in whole bunches can develop more sweetness. So we had two explanations of sweetness in dry wines. First, yeast autolysis, then compounds coming from solid parts of the grapes, and finally we studied the effect of oak wood. And you know that currently during aging, there is an increase of the sweet perception of the wine, despite the level of residual sugar were the same. And we investigated in this way and we identified two compounds, and in fact more compounds, but two main compounds that were QTT, quercetyl triterpenoside 1 and 2. These compounds were sweet. They were present in oak wood. And if we look at the presence of these compounds in comparison in sessile oak and in pedunculate oak, it was led in collaboration, in partnership with Tonnery Seguin-Moreau. We observed that sessile oak was richer in sweet compounds, in sweet QTT, than pedunculate oak. The difference was huge. And for bitter triterpenes present in oak wood, it was more present in pedunculate. It's a part of the explanation to why we use preferentially Cecil oak for wine aging, and maybe pedunculate more for spirits. And we developed a method, a chemical method, to identify the species according to the presence of these two terpenes. So it was a third answer to the initial question: what are the explanation of the sweetness of dry wines? So yeast autolysis, solid grape compounds, and also oakwood compounds, and another applications because thanks to this method we were able to distinguish sessile and pedunculate oak and to compare the effect of this oak. Finally, I would just say some few words about a new approach that we developed, and I find it very promising. It was developed in the PhD of Delphine Vinstel, defended 3 years ago, and it was complementary to the sensory guided, to the tasting that we made. We made a kind of in vivo analysis, in vivo tasting, and due to a partnership with our friends and colleagues from Centers for Taste and Feeding Behavior in Dijon, they were specialists in the receptors, in taste receptors, and they were able to cultivate some cells expressing the taste receptors, and we submitted our fractions to these cells to see if there is any activation of the taste receptors. So it's a kind of in vitro tasting. And Delphine, in her PhD, submitted various fractions and was able to identify very interesting compounds with a bitter taste and a very high activation of the bitter receptors. So these compounds will be published soon, and it can give an interpretation of the increase of bitterness in oakwood aging. So finally, I would thank you for your attention, and I'm really thankful to all the people involved in this work, to the students, to the PhD students, to the postdoctorants. Their work was very precious, and I'm really thankful to them. So thank you, and I will be happy to answer your questions. And now I will let the floor to Marie. She will give you more insight about ISTLB. Thank you very much.